Okay, so before we get going, I graded a couple of the week 1 assignments. They're excellent, exactly what I'm looking for. Okay, generally from week to week they'll be similar, and again you may have to dive a little deeper, like with anything, than just what I provide you. So again, the wonders of technology, right, and the internet. So as we move forward, so anyway, if you haven't done week 1 assignment or unit 1 assignment, again, you want to try to have things done within the modules. So I would say by the end of next week, you'd want to have week 1 done. Again, I don't dock for late work, but if it gets to be 3-4 weeks out, then I'm not going to dock you, but I'll probably talk to you and find out, make sure everything's okay. Or if you need any assistance. So any questions on week 1? So I always start with past, present, future. So anything from last week that you're kind of the basic introduction. Again, a lot of that terminology we'll continue to see throughout the semester, and you'll continue to see throughout your lives. Really, you're learning a couple of foreign languages. You're learning Greek and Latin kind of along the way. So some of the terminology is a little bit, disconcerting or overwhelming, as there is a lot of it. Don't worry, just stay the course, and there is a method to this madness, and that's your early foundation. We'll just keep adding to it. I went ahead and I put, albeit a little bit late, I was hoping to get this week's in last weekend, but I think it was on Wednesday, I finally got it in. And I went ahead and put, I think, up through week 9. So your slides and your outlines for the next 8 or so weeks are in there. Try to get even the rest of the semester done this weekend. But anyhow, that'll keep you busy for now. Okay. If you've looked at week 2, may seem a little bit overwhelming, speaking of overwhelming. And that's why I'm here, is to kind of go through what is important out of all of that, maybe, because there are probably about a quarter of all of that stuff that I think I'll spend a little bit of time on. The rest of it is more or less kind of FYI. Again, how deep you dive is totally up to you. I don't want to slight anybody and keep any info, but I also don't want you to feel like you're being dumped with a ton of info either. So this is cells, these are cells and tissues this week, a little bit of skin, but cells are something we're going to discuss, believe it or not, throughout the semester. As we go into every week, we're going to do different body systems. Part of doing each body system is looking at the cellular level first. So one thing we'll see today is that cells are extremely diverse. Depending on what organ we're talking about, is how the cell is structured and function, and how it functions. Okay? So when we talk about an organ, we're really not talking about the organ functioning. We're talking about the cells. And that's where the chemistry, albeit a little bit, comes in to play. The pharmacology comes into play, because ultimately an organ isn't sick or diseased. It's the cells and the cellular processes associated with that organ. Those are where the problems lie. So cells are extremely important, and your understanding fundamentally of cells, and even a little bit of appreciation for cells, is going to go a long way for you. So I know that up to this point, all of you have probably had a biology course, maybe an anatomy course, maybe a chemistry course, but cells maybe haven't been kind of at the top of the list of interest for most students. And part of it is the lack of maybe connecting it to understanding that micro scale and how it all relates to everything that we or any of our animals may be going through. Inflammation of a muscle isn't because the muscle has a problem. It's at the cellular level, and it has to deal with chemistry. So anyhow, I want to spend about the first 45 minutes or so trying to go through 3 and 4. There's a lot there, but I'm not going to spend a lot of time on a lot of it. It's important that you see some of the big 3 macromolecules: carbohydrates, which provide us energy; proteins, which provide structure, as well as some chemistry, some enzymes, and some hormones; and then lipids, or fats, which provide us with primarily hormones and a little bit of structures in the body. So as we look at cells and some of these little structures, we want to think about a couple of things. How do we transport those 3 items from the mouth all the way to the cells of the body? Okay? How do we get the carbohydrate, which is, for the most part, that the cell is going to use, is going to be broken into glucose, maybe fructose, but monosaccharides, 1-chain sugar. Okay? How do we get that into the cell? Okay? Proteins, we relate those to amino acids. And I think all of you have heard of amino acids at some point. And those are the building blocks of protein. It's also what assimilates as we chew and swallow, or as the animal chews and swallows. They're going to assimilate or absorb, break down and absorb those proteins, which have become amino acids, due to enzymes. Digestive enzymes will break all that down and allow the blood to absorb it and deliver it to the cells. So glucose, amino acids, and then fatty acids. Those are the 3 things we need to get inside of a cell: carbs, proteins, and fats. So cells are little versions of you. They have skin, they have a brain, they have blood, they have a digestive system. They require the same nutrients that you do. In fact, you're the one giving them the nutrients. Okay? So that's kind of the foundation and jumping off point for cells. Okay? So let's take a peek. So how are we going to transport them? And what are we going to do with them once they're in there? So we'll see a couple of specialized structures that are going to help us, help the cells work with all of that. And then there's a 4th called nucleic acids. A 4th, so we have carbs, proteins, fats. There's a 4th called nucleic acids that we'll see. And you know that, you know DNA, or you've heard of that. You know RNA, we're hearing more about that lately with these vaccines. The DNA, the NA is for nucleic acids. So that's your 4th big macromolecule. And there are proteins involved with that, but that relates to cell division and cell memory and the nucleus primarily. Okay? So like some of this early stuff is, you know, more or less a little bit of FYI. This first one, we know that cells are the basic unit of living things. And when we talk about organization, we started at the chemical level, but really, when it comes to structural aspects of who we are, cells are that functional unit. Okay? And they're the ones that are consuming that chemistry. Okay? And then we'll see cells put together make up tissues. Okay? Cells, again, they're responsible for a multitude of functions. Again, we'll talk a little bit about these, but you know, we're not going to go into extreme detail of some of the functionality of cells. We could spend a whole semester on cell physiology. We don't offer that class yet, but I don't think anybody decided that. But anyway, okay. So the key word there is homeostasis. Okay. All roads lead back to that, right? Homeostasis is your body's ability or a creature's ability to maintain some sort of equilibrium or balance. Okay. And that equilibrium or balance does have some ranges. Okay. So cells can be flexible. They can withstand certain small temperature changes. They can withstand small pH changes, but for the most part, homeostasis is the name of the game. Respiration simply means exchange of gases. Okay. The other thing cells need besides the stuff, right? They need to eat, but they also need to breathe, right? So when we breathe oxygen into the body, we're actually hoping that that oxygen gets delivered to the cells. Okay? When we talk about glucose, we also talk in the same breath about oxygen. Those two items are going to go together for energy. Okay? Cellular energy involves glucose and oxygen. Okay? And we'll look at that in more detail later. Okay. Metabolism, that's a word all of you have heard at some point. Metabolism is really just a fancy word for chemical reaction. Any chemical reaction is metabolism. Chemical reactions can either be building something up, we call that synthesizing, or breaking something down. We call that decomposition. Okay, so we'll see that a little bit as well. That metabolism is the are what you eat discussion, right? You eat the protein, you eat the steak, your body is going to break it down into a bunch of different amino acids. Then your cells take those amino acids and string them back together into protein. So that steak becomes your hair, or that steak becomes a piece of keratin or a muscle fiber. Okay. Genetic material, of course. And diversity, not only in appearance, but certainly in functionality. This bar just does not, there's just no good place for it. You can't really get rid of it. Covers up the captions. All right. Evolution of cells, again, we're not going to get too deep into that right now. We do talk about evolution and adaptation at the end of the semester, so we'll kind of come back to this shortly. But yeah, 3 billion years ago, pretty old, right? Long time ago. Okay, prokaryotes and eukaryotes. Again, it's more or less FYI. U means true. EU at the front as a prefix means true. Carry, it has to do with corn, like a corn, it means true kernel, basically. The kernel being the nucleus. Okay. Those are what we're going to focus on this semester are eukaryotes, right? We're dealing with the 5 big vertebrates. Okay. We're not going to look at amoeba or anything too small. All multicellular organisms. That's our focus. People aren't bringing planarians or amoebas to the vet, so we don't need to worry too much about those at the moment, anyway. Cell theory, again, this is just history lesson. I would, you might see it, one of your from the test bank, you may have a question that pops up on that, but again, not that terribly important. Cells, extremely tiny, of course. They range from 10 to 30 microns in diameter. All right, well, let's get in. Those are just kind of FYI, get the ball rolling. Now, cell membrane is where we typically start. Cells are going to have 3 features. Again, we're talking about eukaryotic cells, okay, animal cells. You're going to have a cell membrane, and we see that it says plasma lemma. Lemma means membrane. So we're going to see lemma, again, we're going to see neural lemma when we talk about the nervous system. So it's the membrane of a neuron. We'll see sarcolemma. Sarco means flesh, so we think of that as muscle. Sarcolemma is the covering of, or the cell membrane of a muscle cell or a muscle fiber. So again, as we go, like next week, we get into bones and muscles. So we'll be talking about cells right away. We're talking about bone cells and muscle cells. We'll spend a lot of time on it, but gives you the foundation of what you need for building the big picture. All right, so cell membranes, that's basically the skin of the cell. Cell membranes generally we'll see are they're permeable. They better be, right? The whole point is to get things in, get food to them. We also have to get the garbage out. So it's groceries in, garbage out. Okay? So they have to be somewhat permeable. They're selective, though, about what goes in. Okay? And then cytoplasm, that's everything inside of the cell. Cytoplasm, except for the nucleus, that gets its own category. Okay. So 3 things, that's it. Cell membrane, the cytoplasm, and the nucleus. Now, within the cytoplasm, there'll be kind of 3 subcategories that we're going to focus on. Cytosol, which in essence would be kind of like the plasma or the blood of the cell, that's the liquid. Cytosol is the liquid inside of the cell. Some cells are 85% cytosol. Some cells are closer to maybe 60% cytosol. Okay? So cytosol, that's the liquid portion of the inside of the cell. The cytoskeleton, that sounds pretty straightforward, right? Cytoskeleton, that's probably the framework of the cell, provides some structural aspect, also provides a little bit of I guess, mobility for the cells. Cells are able to move around a little bit for some cells, and the cytoskeleton can help with some of that as well. Most structural aspects on the micro scale and the macro scale are going to be protein based. So cytoskeleton, we'll see some of the material that make up the cytoskeleton is going to be protein based. So again, when we consume protein, breaks down into amino acids, some of the synthesis or building back of the amino acids into protein, sometimes the cell is just manufacturing new pieces of its own cytoskeleton. So some of these beads that the cells are creating, or these necklaces or these chains, are for keeping inside of the cell. Some of it's for delivery out of the cell. So in other words, the cell can kind of repair itself. It can change what it can change its shape based on what its needs are. Okay. So lastly, under cytoplasm are the organelles. Some of these organelles you've probably heard of. I think maybe all of you have heard of a mitochondria at some point or seen that term, mitochondria. That's an example of an organelle. Golgi, the Golgi apparatus. So we'll look at that in a few minutes. But those organelles are going to be what are working with those 3 big macromolecules, carbs, proteins, and fats. Okay. Organelles. L on the end of something means little. So organelle means little organs. Okay, little organs. And then, of course, the nucleus, the brains of the outfit. Okay, so let's look a little bit at the cell membrane. So we're going to go through each of these briefly and give you some structural background of some of these cell features. Okay, the cell membrane, again, generally is going to be flexible. We don't need rigidity. And flexibility comes from hydration, certainly. So we want to make sure that we're hydrated, that the animal's hydrated. Okay. So it's the barrier between the inside of the cell and the outside of the cell. We call that intra, I-N-T-R-A, intracellular. Intra means inside, intracellular. So we'll see things like ICF, abbreviations. That means intracellular fluid. That'll come into play later as we talk a little bit about some of our metabolic principles. Okay, cytoplasm, the inside, intracellular, and then outside the cell, we call that extracellular. Extracellular, extra like exit, okay? Outside the cell. So a lot of the extracellular environment is composed of fluid as well. Okay? Extracellular fluid. Okay? And cell membranes are primarily going to be phospholipids. It lists protein first, but certainly very important. And we'll talk about why in just a second. But phospholipids. Just focus on the lipid portion again. So again, we're looking at carbs, proteins, fats. So fats provide some structure as well, primarily cell membranes. So you always want to relate cell membranes as being lipids. And it's a double layer of lipid too. The proteins that we'll see relating to the cell membrane, those are basically kind of like the doorways. They're the channels. Those are the pores. Those are the means in which we can transport things from outside the cell to inside the cell. Okay? Even water, right? Water is not lipid soluble. Lipids aren't water soluble. So water has a hard time dealing with fats, and fats have a hard time dealing with water. So the cell membrane, because it's lipid, the inside of it is the inside of the membrane, not the inside of the cell, but the inside of the membrane, because it's double layered, is hydrophobic. So how do we get water from the outside of the cell to the inside of the cell? We just make a protein channel. Call it a pore. Even gas, some gases can just diffuse past that membrane. But just about everything needs to have some sort of channel or a pump to get it from inside the cell to outside or vice versa. Okay, so there's a lipid bilayer. So it's showing here the cell membrane, these little, I think there's a way, it might not be on Zoom. I think it's maybe with my slideshow that I can turn this into a little red dot. But anyway, you can see this is the cell membrane. These are these little tails. These are what they call fatty acid tails. So you can see the inside of the membrane, that's lipid based. So water can't really do anything with it. So we have a little pore or its channel. Now, so these are going to be protein based. Protein. This little guy here is showing that this is a carbohydrate attached to a protein. Call this like a glycoprotein. So this would be there. So this is outside the cell. So this might be there for communication purposes. Communicate with some of the items that are floating around outside of the cell. Okay. Again, more of the same. Lipid soluble molecules or materials can pass through the membrane. So like oxygen, carbon, so gases, that's about it. Okay. Oxygen, carbon dioxide. Everything else needs some sort of channel or a pore. And again, it's these globular proteins. So they kind of, your book, and again, most books, they really kind of beat a dead horse with some of this. Again, just remember the cell membrane is primarily lipid based and it's a double layer, but embedded within that we have proteins. These proteins, many of them are called integral. If they're integral, that means they're embedded in the membrane. We have some peripheral proteins that are bound to either the inside portion of the membrane or the outside portion of the membrane. So both of these that they show here are integral. You can see they integrate fully into the membrane. All right. Whereas like this little guy here, or this little guy here, are not integral, they'd be more peripheral proteins. Okay. Again, glyco, the idea is if you ever see glyco or gluco, it's relating to carbohydrates, right? It's relating to sugar. So there, if you see glycocalyx, that means there's some sort of sugar connector that's going to be associated with a protein. Okay, so it's basically a sugar coating that covers the surface of cells. That helps with, what do you think? When you think of sugar, you think of maybe kind of sticky. So oftentimes when the body primarily uses sugar for energy, but there are a couple other spots where we can actually use it to our advantage, and that's as a sticky substance to kind of adhere cells together. That's what that glycocalyx can do. And again, this fluid mosaic, what that's trying to show is that it's basically, you know, a mosaic is a series of different patterns of materials, of structures, all kind of meshing and connected together, a mosaic, and fluids implying that it's not rigid. So cell membranes get what's called a fluid mosaic model. And all that means is that there's some fluidity with the cell membrane. Makes sense. We don't want it to be a rigid, hard brick. Okay. So it's primarily water anyway. And mosaic's just, again, implying that it's more than just one flat kind of single structure involved. It's more than just a phospholipid bilayer. There are a bunch of other little items involved. That's what gives it that mosaic look. We're not gonna talk about caveoli. Flagella and cilia, again, just touching on a couple of things associated with the cell. We're still on cell membrane, but these are associated with the cell membrane. Flagella, typically, it's pluralized, it means tail. This would be tails. Flagella are tails. One would be a flagellum. We take the A and put a UM, a flagellum, and we would see that on like a sperm cell. Helps with locomotion, but very few cells beside the sperm cell, I don't know if there are any that really have a flagellum. Okay, so that's pretty much all you need to know about that. And then cilia, those are little hair-like projections on a cell membrane. They're kind of like bristle brushes on a cell membrane. And they certainly would provide some locomotion, but they help a lot with kind of cleaning things up, filtration aspects. Okay, so cilia, you always want to associate those as being something to do with filtration. So the lungs do a lot of filtration. Okay, so we see a lot of cilia there. And yet they're much shorter than a flagellum. Okay, so let's go in the cell. So that's the cell membrane. Let's take a quick peek at the inside of the cell. Again, we called all of that cytoplasm. Okay. And the primary components of the cytoplasm, again, are going to be the liquid. We call it cytosol. And then the cytoskeleton. And centrosome we'll see as part of the cytoskeleton. That really comes into play during cell division. So I put a little X there, a little dot. Probably out of all of the stuff we deal with the cytoskeleton, the centrosome is going to have these little tubes that we'll see go to the poles of a cell. They kind of go to the opposite locations of a cell. So we'll look at centrosome in a little more detail as part of the cytoskeleton. And then, of course, the organelles, all the little organs. And then inclusions. We'll just touch briefly on inclusions. Think of inclusions like gemstones and jewels and stuff like that, right? Right. So cytosol, again, that's the fluid of the cell. It's primarily going to be made of water, but what's in that water? Well, you're going to have some dissolved electrolytes. What's an electrolyte? Basically a mineral, right? It's like a charged mineral. So like sodium, potassium, magnesium, and those are all electrolytes. Let's see, of course, amino acids, right? Well, remember, those are the building blocks of protein. Simple sugars, again, those are the building blocks, if you will, of carbohydrates. So we could call these like monosaccharides or simple sugars. Okay? And again, amino acids. And then proteins, like full-on proteins that have already been assembled, but usually in the form of enzymes. And an enzyme is some sort of a chemical. Okay? When we look at the body, any body, okay, there are really just a couple different chemical classes. Enzymes, that's a big class of chemicals. When you see a term from here on out that ends in A-S-E, ACE, that is an enzyme. And enzymes are protein-based. They speed up a chemical reaction. Without enzymes, it would take a long time to break down a product or to put a product together. The enzymes. Let's think of any other big chemical groups in the body. One's a whole system. Pretty much regulates everything that you do. Hormones, right? Hormones. Those are other types of chemicals. Okay? So pretty much enzymes and hormones, and then we'll see what are called neurotransmitters. Right? Those are the three kind of big chemical groups. Okay. Okay, and so that's the cytosol, the liquid. Cytoskeleton, again, this is kind of the framework of a cell, gives support and shape, enables a little bit of locomotion. So you'll have some of the cytoskeleton is actually attached to kind of the inner aspect of the cell membrane. So they can shorten and kind of pull the cell membrane and shrink it, so it can fit into different places. It anchors some of our organelles. We want to keep anchored within the cell. And these are the three different types of cytoskeletal fibers. They're in order from smallest to largest. The smallest fibers of the cytoskeleton are microfilaments. They get micro, of course, means small. So this just means small filaments. We'll see this again when we get to muscles next week. We're going to look at muscle cells a little bit. And a couple of the primary components of a muscle cell are going to be what's called actin and myosin. Okay, these are microfilaments, okay, muscle filaments. So again, remember, cells are very specialized. So a muscle cell is going to have a lot more microfilaments than maybe, say, a lung cell or a spleen, or a pancreas or something like that. Okay. Intermediate filaments, just that. They're a little bit larger. Sometimes you'll see kind of bundles of microfilaments will form an intermediate filament. So I could kind of bundle these together and get an intermediate filament. I can also take these filaments and I can kind of make like a tube out of them. So that you can kind of see that going on down here. You got kind of a tubular microtubules. And again, they form a tube, so these might help with transport. We might be able to get something shuttled inside of that tubule. They help with, we'll see microtubules playing a role with mitosis and cell division, which brings us to the centrosome. Again, this is another aspect of the cytoskeleton. Typically find the centrosome near the nucleus, kind of hanging out, basically waiting for the beginning of mitosis, which is called prophase. So the beginning of mitosis or prophase, which we'll see here coming up in a few minutes, involves the nuclear envelope or the nucleus membrane breaking open, and also the centrosome breaking open. Okay. Centrosome is a tiny little ball, and inside of that ball are what are called centrioles. They're two little bundles. They look like microtubules a little bit, but they're two little bundles, and these two bundles are going to go to opposite poles of the cell, and then in essence allow for some extra microtubules or spindles to kind of connect and ultimately create an equator for the cell, for the chromosomes to kind of line up and divide. So the centriole is extremely important when it comes to cell division and mitosis. You just want to relate it to that. And there they are, little bundles of joy. Oh, and you see this word peri and then centriolar. Peri, remember, just means around something. Okay? Peri means around. So around the centrioles, basically a little capsule that protects the centrioles until we're ready for cell division. All right, let's move on to the organelles. We've got a handful here that we need to go through. Mitochondria are first. Again, you've probably all heard of those at some point. Mitochondria, those are what they refer to as the powerhouses of the cell. This is where energy gets converted. This is where glucose and oxygen go. Okay. So again, when we think of consuming carbohydrates or sugars, it's ultimately the mitochondria that are going to work with those carbs that we consume in the form of glucose. Okay, mitochondria. And again, the number of mitochondria inside of a cell varies depending on the type of cell. For instance, like a muscle cell, especially the cardiac muscle cell, that requires a lot of energy. You can imagine, right? For us, ours beats about once every second. So heart muscle is going to be probably the organ that has the most mitochondria per surface or per area, per capita. Okay, mitochondria. So you always associate that with ATP. Those are the three letters, A, T, and P. ATP is cellular energy. Okay. So in essence, glucose plus oxygen will give you the maximum amount of ATP or energy. And we call that process that mitochondria go through cellular respiration. Remember, respiration just means gas exchange. We think of it as breathing, right? So what are you breathing in? You're breathing in oxygen. Where does that oxygen go? Well, it leaves my lungs and goes into my blood and gets delivered to all the cells of my body. And then we get what's called, so when we breathe in, we call it external respiration. When we deliver the oxygen, we call it internal respiration. Once we get the oxygen inside of a cell, we can now perform cellular respiration. Okay? cellular respiration. Just simply means the cell is getting oxygen and it's converting oxygen and glucose into ATP. Ribosomes are next. Ribosomes are the little buggers that deal with proteins. So there's going to be a lot of R's here. So ribosomes deal with RNA for starters. RNA are basically kind of the messengers that are bringing the recipe from the nucleus to the cytoplasm, ultimately to the ribosomes. Right? So when we look at DNA coming up here shortly, we'll look at RNA along with that, and we'll see that ribosomes are responsible for basically translating that code and then stringing together whatever the recipe says we're supposed to do. So the recipe might be for a specific enzyme. So make a photocopy in the nucleus, send the photocopy out via a messenger. That's the mRNA. And that messenger is going to bring it to the ribosomes. The ribosomes deal with then synthesizing or making whatever that recipe says to do. All right. So ribosomes deal with protein synthesis, specifically working with the RNA of the nucleus that came from the nucleus, the photocopy of the chunk of DNA. Now, endoplasmic, and oh, by the way, ribosomes we'll see can be free or attached. Free ribosomes are just kind of floating around inside the cytoplasm. So they're traveling inside of this, and then attached ones are with the endoplasmic reticulum. Endoplasmic reticulum, that's a mouthful. If we break it down, endo means inside, plasmic means associated with the plasm, the cytoplasm. So we're inside the cytoplasm. Well, we already know that. We're talking about things inside the cell. So kind of redundant, I suppose. And then reticulum means, in essence, kind of folded, convoluted, a lot of tunneling and tubing and things like that. Okay? Convolution, reticular. So these are folded, membranous, convoluted tubes inside of the cell. We have rough and we have smooth. We abbreviate it ER. So we have rough ER and smooth ER. Rough ER simply means there are ribosomes attached. Lots of rough ribosomes, RNA, protein synthesis all go together. So ER, these in essence are the factories for assembling whatever the product is. When the DNA or the nucleus says, I need this product, usually the ER is kind of like the factory where assembly goes up, especially for larger type items. The free ribosomes can create things outside the factory. But when we need a little more bulky items, we use the rough ER to produce it. Smooth ER means there aren't any ribosomes on it. Oh, and by the way, the rough ER, we'll see it's oftentimes kind of encapsulating the nucleus, right up against the nucleus. And you'll be able to recognize it. It has a lot of dots on it, the ribosomes. And then smooth ER doesn't have those dots. It's a little more superficial or closer to the cell membrane. It's still inside the cell than the rough ER. Smooth ER does a little bit maybe with some, helps with some of the transportation of the product that's been created. But we also see smooth ER maybe doing a little lipid synthesis. There's some of the fatty acid or lipid synthesis. So carbs, proteins, and fats are all covered. Carbs are mitochondria. Proteins are ribosomes and rough ER. Lipids generally get synthesized by smooth ER. Okay, so all three are accounted for. The Golgi apparatus, we'll see, is basically the packaging center. Golgi Packages. Okay, the Golgi is going to package up a lot of the product that's been produced in the ER. So the factory makes it, then it gets shipped over to the packaging center. Okay, that's the Golgi. Now, how do we get things transported around the cell? Believe it or not, they're called vesicles, which is nice because if you take the S out, it's the word vehicle with an S instead of an H, vesicle. They're little tiny blobs that pick things up and deliver them, to, for instance, to the Golgi. So from rough ER, I make the product. So it's a little protein strand, a piece of keratin or something. I can put that in a vesicle and deliver it over to the Golgi. And then the Golgi can kind of finish everything up, get it ready, maybe store it for a while, right? Just stores it. And then once it gets triggered, then it'll release it. Puts it in another vehicle, and that vehicle or vesicle then will associate right up to the cell membrane, and then it'll, the item will exit the cell. The Golgi also is kind of a waste facility too. So we can see some of these vesicles like lysosomes. Okay? Lysosomes, those guys kind of go, they're like the garbage trucks of the cell. Lysosomes help with cleaning up cellular debris, cellular waste, and they'll oftentimes either deliver that waste right out to the cell, or they'll deliver to the Golgi, and the Golgi will kind of, Golgi's kind of like your colon, kind of processes things, packages it, oftentimes for exit of the cell. Okay. Proteasomes, peroxisomes, they also kind of going along with lysosomes, they help with a lot of the maintenance of the cell's interior. So they help kind of keep the cytoplasm clean. Peroxisomes, especially, you think of like hydrogen peroxide at your home, maybe in your medicine cabinet, but peroxisomes or peroxide is going to help with some of the like antibacterial stuff, maybe antiviral. So peroxisome population of cells, especially important immunity cells, or so immune system cells would have a lot of peroxisomes, for instance. So if you have more compromised immunity, it could be due to a lack of sufficient peroxisomes. Or you have enough peroxisomes, but they're inefficient in what they do. It's not always a numbers thing. It's just, are they performing? So just kind of give you the picture, go back through the mitochondria. Again, they're kind of bean shapes. Yeah. Oh, vaults. Yeah, same deal. They kind of all go with yeah, perfect. Yeah, the vault. Yeah, that's where we'd store some items for future use. Mitochondria, again, they're the largest of the organelles. They're evolutionarily speaking, when we get to the evolution section, I think it's about week 13 or so, we'll come back around to these. These are almost like their own, they're almost like a cell within a cell. There's some train of theory that mitochondria actually existed separately from cells, and part of what enabled cells to be more self-sufficient is getting kind of blended with mitochondria, which then could provide a cell with energy or power. So then that led to other things happening in the water and ultimately on the land. But yeah, mitochondria have their own DNA. That's why they think that. And the mitochondria are like a separate DNA from the cell itself, from the nucleus itself. So just logically speaking, it would seem to make sense that, yeah, they're maybe separate in some form. All right. Because again, they have their own little membrane, they have an outer membrane or cell, it's almost like a cell membrane. They have an inner, like similar to the cell, like a dual membrane, double layer membrane. Okay. They don't have a nucleus though. Their DNA is within the matrix. So anyway, just a little FYI. Just again, associate mitochondria with cellular respiration, glucose, oxygen, ATP, powerhouse. Okay. Organelles, number 2, the ribosomes. Again, these are the most common. So a cell is all cells, most all cells are going to have an abundance of ribosomes. Then they're tiny little specks. Okay. And again, you want to associate those with protein synthesis. And you go again, they make items that are intended for use inside the cell. They make items that are for use outside of the cell for export. They eat, how about a third spot is also the cell membrane, right? There's a lot of integral membrane proteins. Maybe a channel or has been overused and it's just kind of getting a little bit shredded. And so the nucleus gets the message that we may need to fix that door. So yeah, just get the recipe for integral membrane protein X or whatever, and then embed it right into the membrane. So some things are for keeping in the cell, some are for outside the cell, and some are for fixing the cell membrane. And again, ER is just a bunch of flat tubes, a couple different types. The rough ER you'll associate with ribosomes. Protein, smooth ER, think of that as the lipids. And again, they kind of are diagrammatically showing we've got a nucleus, and then we have that rough ER being closer right up against the nucleus, and then the smooth ER. And then this little blob up here is going to be the Golgi. So it kind of goes in order, right? We've got the nucleus. This is kind of the brains of the, this is the think tank. This is the office, where they pull out the blueprints and they decide what they need. And then they send all the directives over here to the factory, and they produce whatever it is that the think tank said to do. And then once they produce it, they send it up to the Golgi, and then the Golgi does whatever it's supposed to do with it. And again, we see the term vesicles, vesicles. Again, you put an H there and you have vehicles. So vesicles are little vehicles that pick things up and deliver them throughout the cell. Some of them deliver it to the cell membrane. So the Golgi packages, Golgi packages, and also distributes, kind of the Amazon warehouse, basically. Or UPS hub. And again, just briefly going through these lysosomes. Again, these are going to be little specialized kind of, garbage men that go around and pick up cellular debris. They'll encase items, and it may not be debris. It could be something that the cell needs. This could be little amino acids that have come in, and so a lysosome may come in and take up some of those amino acids and then break them down into smaller bits. Proteasomes help with, if we've got some sort of a mutation or misfolded protein, there's a lot of checks and balances certainly that go on in cells, because there are a lot more mutations probably than we would want to think about. So thankfully we have a checks and balances, and proteasomes, lysosomes, peroxisomes, they help a lot. Our immune system helps a lot with recognizing mutations and trimming them out. In fact, we'll see when we look at mitosis that when a cell divides and we get it's got two cells kind of going into their life cycle at that point, call it interphase. The first section or portion of a cell's lifespan is just kind of spent observe like a baby. They just don't really do much. They just kind of hang out and observe, and they're kind of getting acclimated, and then they get pulled off the line. We'll see they just kind of get pulled off and kind of kept to the side. They get another checks and balances, making sure that, and maybe we don't need it, so it'll get lysed or it'll get destroyed, go through apoptosis. But if we need it, it'll get put back on the interphase line, heading ultimately toward replicating its DNA and then splitting or dividing. Well, if there's that's where the mutation is supposed to get caught early. If a cell's born and it has a mutation, it probably isn't going to replicate its DNA and be allowed to do that. In fact, the bodies is what the cells themselves are, they're getting plucked out early, hopefully. But, as with everything, environmental factors, genetic factors, all kinds of factors can lead to, a new normalcy, right? Call it autoimmune diseases, a lot of them, right? But your immune system's created like a new homeostasis, new set points, we call it. Your dog's under stress all the time, and their heart rate's always up, their blood pressure's always up. Eventually that becomes their normal. It's their set point. That's not healthy, of course. We could get them back to normal, hopefully. But remember, the body wants to, it desires it. It's just been triggered to be, in a loop. You have to get it out of the loop. Sometimes the medication will do that. Behavioral modifications, usually with the owner, not the pet. Pet just does what it does. The owner is usually the one that needs behavior modification, right? Yeah. So, anyway, some of this info on cellular stuff that we talk about, we're touching on, we'll see it throughout the semester. You'll see it in all of your classes. That's how we have to relate to our patients, well, the owners of the patient, but ultimately they're the patient too, right? The owner. And they may not know any better. Especially now, we've seen with COVID, a huge increase in pet ownership. Some folks, it's like an impulse buy. You're at Walmart, oh man, I need that thing of Altoids, or I need those mini M&Ms in a tube, or I need that little Jasmine keychain or whatever, right? I need a Shih Tzu. That's a big impulse buy. So you're not prepared. So anyhow, inclusions, just those. We've got some little units of metabolic products and substances that the cell is engulfed. We've got, again, these are kind of things that are in the process of being delivered somewhere. We have little vacuoles. These are like mini vacuum cleaners. Vesicles are little vessels, tubes for delivery. Some inclusions are non-membrane bound. Those would be your fats. All right, let's keep on moving. Nucleus, again, it's the largest organelle of the cell. That's why it gets its own category. It's like doesn't get put in with the other organelles. It is the control center. It's the brains of the outfit. And again, this is where DNA is stored. The heredity info. The nuclear envelope is in essence the membrane. Okay? They call it the nuclear membrane or the nuclear envelope. This is the first thing that's going to break open when we start the process of mitosis or cell division. The nucleus has got its own fluid. It's its own form of cytoplasm. We just call it nucleoplasm. Okay, and then chromatin, and then the nucleolus, the small nucleus. We'll see those in a little more detail in just a second. And again, it's almost like the cell membrane. It's got a double lipid bilayer. Outer layer and an inner. This almost sounds like it's 4 layers, doesn't it? 2 lipid bilayers, like 2 double layers. It really should just say 2 lipid layers, maybe, or 1 bilayer. But the emphasis is that there are 2 layers. DNA and RNA, again, we'll spend just a little bit of time. We talk about this further down the line, but I've mentioned it already a little bit, but I certainly want to mention a little, or introduce you to a couple more details. Again, it's the foundation, as you know, of genetic information, and they're different chains of nucleotides. Okay. And these different chains of nucleotides have what we call a nitrogenous base. And these nitrogenous bases are going to pair up. Okay. They're anchored by little 5-carbon sugars. So we'll see that in visual in just a second. But the nitrogenous bases, in DNA and RNA, there are 3 that are the same in both DNA: adenine, cytosine, and guanine. Now, in DNA, adenine combines with thymine. Okay. Cytosine goes with guanine. Okay. But in RNA, we don't have thymine, we have uracil. And they abbreviate all of these with the first letter. So A, C, G, and T. We can do A, G, T, and C, C, A, G, T. Maybe not, I don't know. Probably waste of your time, but anyway, I'm gonna stay on track. I'm trying. Okay. When RNA is a copy of the DNA. You know, when we need to create some sort of enzyme or protein structure, okay, we don't want the entire recipe book. DNA is not going to just give the whole recipe box out to the rough ER and say, well, it's in there somewhere. Just you figure it's under the S's. Find it, right? No. It's going to take the recipe out. It's going to make a little photocopy of just the bit it wants. That's the RNA. Okay. And then the RNA, so the RNA is basically a replica or a photocopy of whatever the DNA says, or the nucleus says we need to create. And so thymine gets removed in the photocopy, and we put a U instead, uracil. It's double helix, so it's basically kind of like 2 necklaces kind of twisted around one another. So we've got kind of like a sugar anchor. The ribbon is anchored with sugars and phosphates. And then again, we can see these base pairs. And they're bound by hydrogen bonds. Again, not to get too deep into chemistry, but a hydrogen bond is the weakest bond of all bonds, which is nice because when I need to make a photocopy, it's very easy to break those bonds. So again, A's go with T. So adenine and thymine go together. So one of these is an A and one of these is a T on each side. They colored them down here. So this would be a T and this would be an A. This would be a G and this would be a C. So C's and G's can only go together. And you can see they're kind of like lock and key fit. And again, RNA is just a single strand. It's the photocopy from the DNA of whatever it is we need to produce. And again, uracil is going to go with adenine. Chromatin, that's the genetic material. It's the substance of the chromosomes. Chroma means colored, so these are a different color to them. So when we uncoil the chromatin, okay, we're going to end up with a bunch of little chromosomes. So these chromosomes are kind of compacted and spun in a coil. It's called chromatin. And the nucleolus, some nuclei have more than one nucleolus, but typically we just see one. Some have multiples. And they're not bound by a membrane. And this is where those ribosomes and the ribosomal subunits are going to be created. So this is like the real storage area. This is where the real action, I suppose, happens regarding the think tank. This is where the DNA is located. There's some floating around, of course, in the nucleus. Again, and we will go through that, all that stuff in more detail. So let's see. We should probably take a break. Probably past that time. Already 20 after, huh? All right. You guys want a break or you want to keep going? A lot to digest, I know. Well, let me just tell you briefly. This chapter, cell physiology, it's pretty heavy in details of all of the stuff we've just talked about. So I don't really spend much time here. The state doesn't require us to go into extreme detail of some of these functions, like cellular respiration. You need to just associate that with glucose and the mitochondria, oxygen and glucose in the mitochondria to get ATP. Protein synthesis. And again, we've got the DNA and the RNA making photocopies. This one talks a little bit about the fluid compartments. Again, we have the extracellular fluid and the intracellular fluid. Okay, so these fluid compartments, so this talks about moving stuff. Okay, so how do we get these electrolytes to move from outside the cell to the inside of the cell and vice versa? It talks about concentration gradients and having these ratios kind of talk about equilibrium all of the time in regards to cell metabolism and cell physiology, maintaining equilibrium. So anyway, generally we think of movement of either stuff. So we either move the solids or the gases. So we'll start with the gases like oxygen, carbon dioxide. They just diffuse based on numbers. So when there's a lot somewhere, they'll diffuse where there are less of them. So oxygen, when we, inside of our bodies, inside of the bloodstream that's coming to our lungs, that bloodstream is low in oxygen. Just exhale the carbon dioxide. So when I breathe in oxygen, it goes from high concentration, which is out here, to lower concentration, which is in the bloodstream. When the blood picks it up, delivers it to the body, and same deal. Inside of the cell, there's less oxygen. We're delivering oxygen to it. So once there's a lot of oxygen outside the cell, it's just going to diffuse where there's less oxygen inside of the cell. Okay? So that's how the gas is. Pretty straightforward. They don't need any pores or channels or anything. The gases can diffuse right through that lipid membrane. So that leaves us with the solids. So there's the solids and liquids. Solids get in the multitude of ways, those channels primarily. And some of those channels have different sizes based on what the cell is used to getting a lot of or not much of. So the cell has specific needs for magnesium or whatever, then it's going to have pores that are just the right size for magnesium. So so that's still diffusion generally. Is a lot of something outside is not much inside, so it's going to diffuse to where there's less of it. So now we have what's called simple diffusion, which is basically what the gases are. Simply a lot of them here, not a lot there, so it's simply going to go there. The other one's facilitated. Facilitated diffusion involves some sort of helper. Facilitator. Something like glucose needs a facilitator, like insulin is a facilitator. There's a special channel. So these channels are like locks. They have a certain key that will fit into the lock to open it up, so we can get whatever it is inside of the cell. So that's all facilitated diffusion. Sometimes there's an enzyme outside of the cell that kind of attaches to the membrane protein and then triggers it to open. So they're called gated channels. We see one called ligand or chemical gated, meaning a special chemical has to unlock the door for the substance to get in. We have voltage gated. So some of them require a little jolt of electricity, and then the gate will be a little key card or whatever, an electronic card, voltage gated. So there are different types of gatings for getting things in. So that's simple diffusion, facilitated diffusion. Okay. The third form of moving a substance, a solid into the cell or out of the cell, involves the usage of a pump. Pumps require energy. So pumps only are needed if I'm pushing something against its concentration gradient. So they go in the opposite direction. So moving something from not much to where there's a lot of it. Like trying to shove extra in where there isn't room, but we have to get a pump in order to do it. That takes energy. So we see sodium and potassium are the big dogs for that. And sodium generally lives outside of the cell in large quantities. Potassium lives inside of the cell in large quantities. But sodium, so naturally, right? Sodium, a lot of it outside, not much in. So normally we eat a lot of sodium. Sodium would just diffuse and go into the cell, which it does, but it has special pores that only open up if we get some triggers to happen, which is good because we don't want sodium rushing inside of the cell all the time, because there's always more of it outside the cell. In order to get back to equilibrium, we have to pump it against its concentration gradient. That takes ATP of energy, so that the cell has more requirements. So sodium generally is very regulated how it gets into a cell. Let's see. So anyway, pumps. Oh, the other option. So that's how we move gases and solids. The other thing we could do is move liquid. We could dilute an area. So I've got a ton of sodium outside of the cell. Generally, the body won't open up those little gates to let it go inside the cell because of the pumping to get it back out. It's too much. It's like a dog chasing its tail. So what they do is say, well, let's dilute all that sodium. So the cell will send water from inside, cytosol. It'll shoot water out through the pores, kind of dilute that area outside the cell. That can be problematic over time. It leads to edema or swelling. Because that space outside the cell is called the interstitial space. It's also called the extracellular space. Yeah, if we get too much sodium, then the cells will shrink a little bit and dehydrate and shoot water out to kind of dilute all that sodium, which then triggers the kidney to work harder because we've got fluid osmolarity issues or osmosis issues. So movement of fluid is simply osmosis. And we move fluid from an area where there's a high concentration of solutes. Right? So that's the long and short of this entire chapter, basically, in a nutshell. Osmosis, diffusion, solutes, solids. We talk a little bit about acids and bases or pH balance in this chapter as well. Again, that's something we'll see throughout the semester, so I'm not going to spend too much time on it. I do want to let you know, though, that the H means hydrogen, okay? Little p, big H. It's a big H because that big H represents hydrogen. It's basically like percentage of hydrogen. The smaller the number, okay, the smaller the pH, the more hydrogen ions. It's somewhat counterintuitive. So a pH of 1 is acidic. pH of 1 has the most amount of hydrogen ions. the number, the pH number, and again, it's too early. Well, anytime it's probably too early to talk about this, but it's what we call a base 10 formula, the amount of hydrogen. So it's negative exponents. Negative exponents mean we're dealing with decimals. Like if I have 10 to the negative first power, I have 0.1 whatever. So I've moved the decimal over once to the left. 10 to the negative second means I'm moving the decimal over twice to the left. So it's 0.01. Okay. So pH is determined by that negative exponent. So 10 to the negative first amounts of hydrogen ions means I have a pH of 1. 10 to the negative 7th is a pH of 7. It's all about amount of. Would you rather have a penny? Would you rather have a 0.1, which is a dime, or would you rather have a penny, which is 0.01? Right? You'd rather have a dime. 0.1 is more, and so if I write 0.1 as an exponent value, it's 10 to the negative first power. If I write a penny out in negative exponents, it's 10 to the negative second power. I probably already said way too much about pH scales, but that's where they get all of that. The more hydrogen, the more acidic it is. That's my point. It's like a pH of 12 is 0 point, and then 11 zeros, 1 amount of hydrogen. Very little hydrogen. So they're hydrogen acceptors. They're poor in hydrogen. Acids are hydrogen donors. They have a ton of it. They have 10 cents worth. What's 10 to the 0th power? Can you have a 0 pH? Yeah, you can. It's because 10, any number to the 0th power is 1. So 10 to the 0th power is 1. So it means 1 hydrogen ion relative to, but we think of pH of scale as being 1 to 14. We don't really think of pHs of 0. Okay? pH of 1 to 14 is generally what we think of. Okay, water is neutral, pH is 7. The stomach acids are low. They're like a pH of 1 or 2, like hydrochloric acid, right, in the stomach. Okay, and blood is slightly, what we'd say, slightly alkaline or slightly basic, 7.4. So we'll talk about when we get to the cardiovascular. So we'll circle back to these pH stuff. When we get to digestion, we'll talk a little bit about it again. When we get to cardiovascular, it's big there because we'll do, and then when we get to the kidneys and urinary, we'll talk about pH again. But yeah, the blood pH, we deal with what's called acidosis or alkalosis. Sometimes it's a condition where the blood level's too acidic or the blood level's too alkaline. And there's a range 7.35 to 7.45. And when our blood is slightly acidic, then we will see when we talk about the respiratory system, breathing patterns change. Carbon dioxide allows for very acidic kind of environments. One of the byproducts of respiration in the body is carbonic acid buildup. And the carbonic acid buildup can cause some problems in the lungs. It can cause problems with blood pH. Carbonic acid, it's like adding an acid to the 7.4. It's going to bring it down to like a 7.2 maybe, or 7.1. And when cells are immersed in an acidic environment over a chronic period of time, it leads to mutations, mitotic events, and cancer. We're starting to link cancer with a chronic pH values below normal or in the acidic range. So we'll talk more about it. How do you regulate pH? Respiratory, you got to exhale to get rid of carbon dioxide. So sedentary pets, sedentary humans, sedentary anything, leads to respiratory not doing much, leads to a buildup of carbon dioxide, leads to pHs, more like acidosis, called metabolic acidosis or respiratory acidosis. So then the kidneys kick in and they eliminate hydrogen ions. Remember, too much acid means too much hydrogen. The kidneys regulate hydrogen ion concentration, so then you'll pee out, or your animal will pee out a lot of hydrogen ions. So they pee more. So frequent urination could be a sign too that there's some pH issues. Okay, electrolytes, again, we're talking about calcium, potassium, magnesium, zinc. Okay, the stuff in Gatorade or in Powerade, you know, these energy drinks. No, I don't know about energy drinks. That's like Red Bulls, but I guess like the refreshing drinks that have they even have water now, right? Infused with electrolytes. Okay. Again, we're not going to go through this today at the moment. Kind of talked about it already, the movement of fluid. Remember, always remember, water moves to where there's more stuff. Water follows sodium, basically. So if I have a solution with a ton of sodium in that solution, the cells are going to release water into that environment to dilute all that sodium. Okay, that's what we call a hypertonic environment. The tonicity deals with the actual solution, not the cells. So hyper means a lot. So hypertonicity means I have way too much stuff inside of the fluid, the extracellular fluid in the solution. Okay. Iso means same, so that's normal. We've got the equilibrium. Okay, so the cells won't shrink or rupture. Okay, so hypertonic, again, the cell will shrink. Hypotonic, the cell will blow up. And edema, I mentioned earlier, again, is, you know, that movement of water into the extracellular interstitial space to dilute excess amount of sodium, typically. And again, you know, you could go through and kind of look through some of this again on your own if you'd like, but it truly, the stuff I wanted to discuss, we've really pretty much hit on already. So let's see. So osmosis, simple diffusion, facilitated diffusion, filtration, these are all passive processes, what we call passive transport. Passive means they don't require energy, means or ATP, means they just happen because of laws of physics. Okay. Excess here moves to where there isn't as much over there. Okay. Now, active transport mechanisms require ATP. Think of active transport process, ATP. That's not what ATP means, but it's a chemical. Adenosine triphosphate is what it means, but we're not worried about that for now. Just know that active transport means I need ATP. It's going to come in the form of a pump. It can also come in, going back to, remember we talked a little bit about vesicles and vehicles delivering things to, they also need ATP. So that's another type of transport that occurs between cells that requires ATP. So active transport, primarily we think of pumps in the membrane, pumping against concentration gradients, okay, pumps. The other one we think of, again, would be any of those little transport vesicles. Think of a vehicle needs gasoline. Okay. So ATP is the cellular gas. Oh, so that's what cytosis means, is that transport. We have what's called endocytosis, and that means bringing something into the cell, and exocytosis, something exiting the cell. The vehicles do all of this for us, and they need ATP. Kind of fuel up the FedEx truck. So endo means bring something in. Phago and pino. Phagocytosis means solids, like bringing in, so eating basically. Phagocytosis is eating. Pinocytosis is drinking. So bringing in liquids versus bringing in solids. So solids are phago, liquids are pino. Again, a lot of this type of stuff is going to involve the immune system. So, when you have little, microbes floating around in the plasma that aren't supposed to be there, the body gets triggered and white blood cells respond. And a lot of our white blood cells are going to engulf these bacteria or these viruses and consume them. Okay. Gases just kind of move based on diffusion principles. They don't typically have any type of transport helpers or carriers. Yep. They don't even need a channel, gases. Well, when we get to tissues, we'll see that there are specialized tissues for gases, and they're always very thin layered to move gases quickly. Gas transport is the speediest of all transport. And so what we see is very thin membranes for gases to just move right through. Yep. And all based on diffusion principles, simple diffusion for gases. And the primary gases really are going to be oxygen and carbon dioxide. We see some hydrogen as well, but oxygen and carbon dioxide are the primary gases that we'll deal with throughout the semester. And again, oxygen in provides the one ingredient for the recipe for ATP, and CO2 out. And when we get to the respiratory system, we'll go through a little bit of the physiology and exactly what's going on with that oxygen. So we'll talk a little bit about cellular respiration again in mitochondria, and then we'll talk a little more detail about pH levels and the importance of eliminating CO2. The way the body functions is absorption and assimilation, extremely important, right? We need to absorb and assimilate nutrients, but excretion and taking out the trash, just as important, equally. Talk about eating the right foods. Well, we also need to talk about pooping the right poop, right? It's part and parcel. We don't want to hoard garbage. It's like at home, right? Groceries in, garbage out, not just groceries in, groceries in, groceries in, groceries in. We never take the garbage out. That causes stagnation and clogging and ultimately inflammation. So, but gases, yeah, they have an easier time of movement. But some of the byproducts of carbon dioxide not getting moved out fast, or as I guess efficiently as it should, that leads to buildup of carbonic acid, some of the byproducts of respiration. And these lead to hydrogen ion concentration increases or pH increases. This next area talks about basically the charge, the potential for there to be action at a membrane. So we're dealing with ions or ionic. An ion is a charged particle, a charged element. An ion is charged. Okay. These are your electrolytes. Okay. Sodium and potassium, magnesium. And again, normally more potassium inside the cell, more sodium outside of the cell. Those ions then create an electrical charge. We call it, when we're at rest, we call it a resting membrane potential. Potential simply means that something might happen, right? So, but we're at rest. And when we'll see this and talk about this in more detail, especially when we get to the nervous system. So in about 2 weeks, we're going to talk a little bit more about ion movement, what's called RMP, or resting membrane potential. Then we'll get into what's called threshold, and then hitting what's called an action potential. And then that's going to cause a flood of ionic movement. And so again, I don't want to get the cart too far before the horse, but I do want to at least introduce you to the fact that sodium and potassium exist, and they kind of exist together. They're kind of like opposites, though, of one another. They're antagonists. But anyhow, in order to get energy, again, in order to get action to happen, some sort of movement to occur, we need to alter ion placement, and that's going to trigger then things to happen. So right now at normal resting membrane potential, we've got a certain amount of sodium outside, certain amount of potassium inside. If I need action to happen, then I start shuffling these ions, and that's going to trigger some different things. So anyway, that's way down, not way, it's a couple weeks away. Okay, cell division. I don't think we're going to make it to tissues or skin. So here's what I'm going to do today. Yeah, we're running. So my next class, I'm not going to talk about any of this. I'm going to talk only about tissues and skin and record it, and then you'll have to watch that recorded. And they're not going to hear any of what we've done today. They're going to end up having to watch this one. So life cycle of the cell. So a couple of key features with this. I'm trying to make this palatable. It's not as bad as it's made it seems. The bulk of the cell's life, probably 85% of the, and it doesn't really represent it well there, but it's actually more. 85% of the cell's life probably is spent in interphase. Interphase, inter means between. Phase just has to do with, it means turning points or something happening. So, so from basically birth of the cell till it's ready to die. So instead of dying, though, it's going to divide. So from birth to death, in essence, that's called interphase. That's the bulk of the cell life. So interphase is 3 phases. G means growth. So G1 is the first growth phase. This is what I was talking about earlier, where it's kind of a newborn. It's not really doing much. It's kind of observing its environment, getting some directives of just kind of staying around, hang out. Well, during G1, it doesn't show it, but right here, every cell gets pulled off the line. Okay, so once it's ready to go, maybe off to high school or something. Made it through 8th grade, ready to graduate, we pull it off the line into what they call the GO phase or the G0 phase. Okay? And what that means is we're kind of putting a halt on the growth of the cell. It's put into quality control. Some cells are put into storage, in essence. There's just not a need for it, but it's perfectly fine. Quality control says the cell's healthy. We just don't need it yet. So it just gets kind of set off to the side. That's going on. Oftentimes during GO phase, the cell will die. It'll get put through apoptosis. Remember, one cell is here. When I get here, I've got 2 cells. So I can't allow both of these cells to thrive. Because both of them, if they both thrive, then they create 2 cells. Now I've got 8 cells. You know, things get wild. So one of these is going to, maybe both are going to, and that happens at the GO phase. But if we need it, or if it's healthy or whatever, then we put it back on the train. And it's in interphase. Now it's in S phase. So this is kind of like it's gone off to college, and it's, you know, it's got its degree, it's ready to go to work, and so it makes a copy of itself. It replicates. So S phase is when it makes a copy. So this is where you can see, to prevent mutations from continuing, this GO kind of rest stop or pull off is extremely important. And also recognition, cell recognition. Sometimes cells will recognize a mutant as being okay. Sometimes cells will recognize normal cells as being mutants. So recognition occur, that's important. So anyhow, we can eliminate a lot of mutations from happening by pulling them off the line and leaving them on. So because we're making a copy at this point, if it's a mutation and we let it go, now I'm copying the mutation. And now I'm going to keep removing, and now I'm going to end up with two cells that are identical, but they're mutated. All right. So anyway, once we go through S interphase, we get to kind of the twilight years, head to retirement, G2 growth and final preparations for I guess the big day. So once we end interphase, we're going to see that centrosome break open a little bit, and the centrioles start to go to the poles of the cell or opposite ends of the cell. At the same time, the nuclear envelope is breaking open. That's allowing chromosomes to kind of swim around now in the cytoplasm. That's what we call prophase. So mitosis is now beginning. Prophase. Pro, think of that as like I'm all for it, right? So I'm all for cell division. That's the first thing we have to do is open the envelope, let the chromosomes loose. The next phase is called metaphase. At this point, the chromosomes, remember too, those centrioles have gone to the poles, but all these microtubules have formed kind of like spindles. They're almost like lines of longitude on a globe. And then what happens is on that equator, all of these chromosomes line up right down the middle. So M, or metaphase, is the middle phase. They're lined up down the middle chromosome. And then anaphase is when those centrioles at each end, they kind of start to pull a little bit. And all those webs or strings attached to the centrioles pull with it. And what it does is it splits the chromosomes. So the chromosomes are like, they're not X's like this. They're more like X's like this. So they pull apart. Anaphase is the splitting of the chromosomes. And then lastly, they're going to kind of break apart a little bit. We get what's called a cleavage furrow or a cleavage line. And it's kind of a groove in the cell that's now distinctly showing that it looks like we've got two cells. Telo means they're kind of pulling apart at this point, the two cells. And then that's the end of mitosis. So mitosis is PMAT, P-M-A-T, PMAT. Prophase, I'm in favor of it. Okay. Metaphase, chromosomes in the middle. Anaphase, the chromosomes are split. I always remembered it as a banana split, banana split. For splitting of the chromosomes. And then telophase, like telephone, they're like apart from one another. Okay. So PMAT, those are your mitotic phases. And then lastly is what's called cytokinesis. Kinesis means movement. So at this point, the cells actually are separate and they move. Okay? They're ready to go. They're ready to start crawling. So you've got two cells now. And then just starts all over. And every cell of the body or every organ of, all cells of specific organs have different lifespans. So like a keratin cell on the skin is in the epidermis is like 28 to 35 days. So every 28 to 35 days, we're seeing mitosis going on with skin cells. Red blood cells last, say, like 120 days, for instance. So anyway, all cells have a different kind of lifespan. So you can go through and look at and read through some of this more specifically on your own. Already, the last parts of this chapter goes through protein synthesis. Again, we already talked about it. Just remember, ribosomes synthesize proteins. Rough ER. That's it. right now. You can stay if you want. It's at 10:30. Yeah. So yeah, if you want to hear part 2 live, you can stay in here if you want. I know we go till about 12:30. Yep, I'll post them both. Yep. It'll be it'll be probably like 1-ish. Usually takes about a half hour to like convert the file and do all of that. Yep.